Okay, I'm going to try and stay on time. I do have a tendency to ramble, so I'll do my best. Thanks a lot for the introduction, Mark, and the warm welcome to everyone here. I did get caught in the turbulence on the way in myself, so that was a fun ride. But anyways, I'll just get right to the punch. So I'm going to do a crash course today, essentially to make sure everyone's on the same level, let's say, as we move forward into the talks over the rest of today and into tomorrow. There's a lot of terminology in this field and a lot of basics that are really good to just get the groundwork for so that everything else kind of goes smoothly, let's say. Okay. I'm just going to do my disclosures off the bat. My Pfizer one's probably coming to the end because this was a few years ago now. I did some consulting with them. They no longer have neuroscience or psychiatry anymore, so there's very little relevance for me to be even talking to them at this point. We do have, I've done some consulting for GW Pharmaceuticals, who's a company based in the UK that does cannabis-based therapeutics, and we do have some operating funds from them, although it's not related to anything I'm going to talk about today. Okay, so I'm just going to start off by defining the cannabinoid system and making sure that there's a lot of basic terminology that we're all on the same page about. And so this is, when we talk about cannabinoids, it becomes very confusing, I have to say, to a lot of people because the word is thrown around synonymously for a lot of different types of molecules and classes of drugs, let's say. And even if you start breaking things down into the exogenous cannabinoids, people get confused by this because you have essentially two classes of exogenous cannabinoids. And these are, when I say exogenous, I mean these are the ones that are not generated with our own body. They are taken supplementally from an external source. Now these ones kind of come in two flavors, which are either the phytocannabinoids or the synthetic cannabinoids. Most of you are probably more familiar with the phytocannabinoids in this crowd. And this is essentially any of the plant-derived cannabinoids that come from the cannabis plant. Now, obviously, the most well-recognized one in this realm is THC, or delta-9 tetrahydrocannabinol. There's probably, I'd say, number 2, and coming close to getting a lot of the attention nowadays is cannabidiol, or CBD. But in addition to this, there is probably somewhere between 60 to 70 at least other cannabinoids for whom the biological activity is largely unknown at this point. And that's in addition to a host of other terpenes and other molecules that are found within cannabis that can theoretically modulate some of the effects of THC or even CBD. So those are the basics for phytocannabinoids. Under synthetic cannabinoids, this becomes also more confusing, and I didn't realize this until I started giving some public talks and people were making comments afterwards because a lot of the data, if I give more of a research-based talk, relates to clinical data, and a lot of that in this sphere comes from the use of drugs like nabilone, which is essentially a synthetic THC. Now, if you say synthetic cannabinoids, because of what's happening in the general public with these other synthetic cannabinoids, people start to confuse them. So it's important to recognize that there are kind of differences when we talk about synthetic cannabinoids. There are the kind of medical grade ones, which are standard, like nabilone, dronabinol. I don't know how redundant these are, if this is just different names. There's Sesamit, Marinol. But then there's more so the synthetic cannabinoids, which are the ones that are hitting the streets. And what these are, is these are drugs that are being made in a chem lab somewhere around the world. They get shipped to the street, and they're usually based off of synthetic analogs of other CB1 agonists like THC, for example, that are made in a lab that have a high efficacy and are strong binding potential at cannabinoid receptors. And so these have turned things like K2 or spice. We've learned actually a lot about the differences between gross activation of CB1 from a synthetic agonist versus THC, because one of the things that we've seen with these synthetic compounds that we haven't seen really with consuming cannabis is these overdoses or fatalities that have been associated with it. A lot of the synthetic ones like K2 or spice have actually been related to cardiovascular responses that have resulted in fatality. So that's, to me, a very interesting fact that people have found that. And I was always, frankly, confused as to why these drugs even hit the market. Because of the fact that there is a plant like cannabis that makes THC that seemed readily accessible, you know, for the most part around the world, and for generations people have been using it. Although someone pointed out to me in the States, one of the reasons these synthetic cannabinoids have been on the rise is because people don't test positive in drug tests. And I was quite surprised to learn that, but it kind of makes sense, actually, that people would choose an alternate that they thought created a similar response to them, but they weren't going to fail a drug test. Now, the danger associated with that is obviously what we've seen, which is that there are some physical consequences to these drugs. And so as we move into legalization, certainly one thing I'm hoping to see in Canada is that those get kind of killed out of the black market because they've proven to be kind of dangerous substances. So those are the classes of exogenous cannabinoids that we deal with. And then there's the endogenous cannabinoids, and this is what I study for the most part. I'm a neuroscientist who studies mostly the biology of the brain. And so we call these the endocannabinoids. These are the ones that our brain makes naturally. These molecules have some pharmacological properties resembling THC, but they're not really the same way. And the two that are kind of very well established and recognized in the field, and the ones that we tend to spend most of our time studying, are called anandamide and 2-arachidonoglycerol, or 2-AG. And these molecules have some differences in binding efficacy and affinity for the cannabinoid receptors, and they seem to subserve slightly different roles in the brain and the body. I'm not going to go into that in too much depth, but I think it is important to understand that our body does make these molecules. I always try and liken this to the endorphin system. We have naturally occurring molecules. It's not as if cannabinoid receptors were kind of evolved into our bodies in the hope that one day we would find cannabis and therefore have a cognate ligand for it. This is a naturally occurring system in our body. And as I'll talk about later on, it's interesting because we've learned a lot about what the biology of the system probably regulates in terms of physiological processes just from what we can glean from the effects of cannabis use in humans. Okay, and then as I'll go into in a bit more depth as we move along, is the receptor targets. So these drugs, both the endocannabinoids and the exogenous cannabinoids, for the most part, most of the physiological effects they exert on the brain and body are mediated by activation of specific cannabinoid receptors, which I'll go into a little bit more depth on, which is CB1 and CB2. So this is, in many ways, I find this field turns into an alphabet soup. There's just a lot of letter number abbreviations everywhere, so it's hard to stay on top of. We'll see. Hopefully you can keep up with all of it. Okay, so I feel like it's always important, if we're going to see where we're going, it's important to see where we've come from. And I'm not like, I actually don't have any interest generally in like history of medicine or neuroscience, yet I do find the history of cannabis and cannabinoids quite a fascinating area because it's had several important key inflection points where a discovery was made and it had a huge impact on the entire field and it changed the way that we move forward with a lot of things. So I'm going to highlight a few of these just so that everyone's on the same page with what we've learned so far about this and where we're going with it. Okay, so the first one was the discovery of THC. Now, small known fact that a lot of people don't know, the original paper that characterized THC was published on April 20th. In 1964. There is a running joke over whether this is where the whole 420 thing came from. I would be quite surprised if that is at all true. I think this is just a happy coincidence, let's say, that the original publication characterizing THC came out on 420. But it did indeed. And I remember it was actually a few years ago at this meeting I learned that from John Page when he was talking about it, because in 2014 it was technically the 50th anniversary of THC. So this was discovered by Rafi Meshulam, who is still kicking it in Israel at the Hebrew University. Rafi has got to be well into his 80s at this point, but he's still an active figure in the field. And Rafi's a medicinal chemist, and they had been extracting components of cannabis and trying to figure out what did what. And essentially, in this original publication, what he was able to identify was that THC was the primary psychoactive constituent of cannabis, and he was able to isolate the molecule. So that then gave the field a working map, let's say, to work off of. So now we had a molecule we knew was the main active component of THC. This was similar to when people would identify out of poppy seeds that opium was the active molecule. So you start to understand the biology, the pharmacology. You can study the molecule in more depth and figure out what it's doing to the brain and body. So after this discovery of THC, though, it was very important in the field for us to identify this. But the big question that came about after this was, well, how does THC work? Now, THC is unlike a lot of other drugs in the sense that it is a lipid-soluble, it's a lipophilic compound, and that kind of was different than what we saw with the majority of other drugs, which are more like, you look at things like cocaine or morphine or alcohol. These are not lipid-soluble substances. The way that they interact with neurotransmitter systems was a little bit easier for people to figure out in this era, let's say, with the current state of technology at that point. So there was a lot of questions about how is it that THC is exerting these effects. One of the things I will say up front that I find fascinating is as we get into the biology of the system, it is extremely widely expressed throughout the brain and body in a ton of different tissue systems, and even in the brain on a whole bunch of different circuits throughout the brain. And what's amazing to me is that a drug that has such a ubiquitous biology somehow has a relatively consistent phenotype when it hits a system. So I find that quite fascinating. And in the early days, there was some recognition of this as well, and this kind of guided the way that they thought about the possible mechanisms of THC. So one of the very early theories actually related to the fact that if you looked at the structure of THC, the thing that at that point in biological systems that it most similarly represented was stress hormones or glucocorticoid hormones, especially cortisol. And so there was this original theory that actually THC was essentially a false steroid. And what it was doing was, because of its structural homology to molecules like cortisol or estrogen or progesterone, that a lot of its biological effects were just going to be mediated by this indiscriminate activation of hormone receptors throughout the body. And this was, as I said, this was also interesting from the context of people making these theories, because of the physiological effects of cannabis in some ways were resembling some effects that had been documented with steroid hormones. So if you gave someone cortisol, or at least a medical version like hydrocortisone, you saw a lot of physiological effects that were similar. There was pronounced anti-inflammatory responses, which are definitely stronger with steroid treatment than they are with cannabinoids, but same direction. There was some orexigenic effects. People tended to eat more. One of the reliable consequences of going on steroid treatment is people do tend to gain weight. They become hyperphagic. They retain water. They have shifts in metabolism. And there were a lot of reports of euphoria, especially people going on high doses of steroid treatment, that they would actually have euphoric responses. I remember years ago when I was in grad school, I had a random virus illness, and after I was kind of recovering from this, they put me on this high-dose steroid treatment. I remember being braced by the doctor. You might feel some euphoria, and I was like, please. At that point, I was in a very bad state, but I didn't get any of it, so I was very disappointed, I have to say. They shouldn't say that to people. But it was really interesting, because this was kind of one of the original theories, is that this is really how cannabinoids like THC were working, was through indiscriminate interactions with steroid systems. Now, there was actually some evidence to support this. And the unfortunate thing was, as we discovered more about the cannabinoid system biology, this area was abandoned. And I think that there's a lot here of relevance, and I'm kind of glad to see the field picking this stuff back up again, and even my labs developed some interest in this again, because there was evidence back in the 70s and 80s that molecules like THC and possibly cannabidiol or some of the other cannabinoids had the ability to directly interact and bind with steroid receptors. And so you could remove, for example, adrenal glands and take all the sources of cortisol out of the body and administer THC and still see binding responses at those receptors. So there's something going on there that's not being fully appreciated. But the fact of the matter is, as the field moved forward, this theory was largely abandoned because it couldn't explain a lot of the effects, and there was some inconsistencies. Fair enough. Okay, so then we moved into the 80s, and this is when neurophysiology was taking off a lot more in the field, so people were using techniques to be able to look at neural activity and other approaches. And one of the things that had become quite apparent at that point was the influence of cell membrane fluidity on the functioning of ionotropic receptors, or ligand-gated ion channels, essentially, in the brain. So a lot of our fast-acting neurotransmitters like glutamate and GABA exert their effect by activating these ion-gated channels that regulate ion flux across a membrane. Now, at that point, people had started doing studies where they were doing depletion of cholesterol and other approaches, and showing that if you modulated the membrane fluidity of a cell, you modulated the manner in which these ion channels were functioning. So this kind of led to the idea then, again, because of the lipophilic nature of THC and the fact that it had a tendency to embed itself in lipid-friendly environments like a cell membrane, that basically what would happen is, if someone consumed cannabis, you would get accumulation of THC or other cannabinoids in the cell membrane, and that would then modulate the fluidity of the membrane itself, and that in turn would then change the ability of ion channels to function. Now, this was kind of put forth in the field as a theory because of the fact that it would explain these kind of broad nervous system effects that were found with cannabinoids, and perhaps it was having specific influences on some aspects of ion channels, and that's why you had specific changes in, say, feeding and pain and sleep. But the issue, again, was this was really nonspecific in nature. There was a lot of things that didn't hold up to, so this theory wasn't really held onto too strongly, but it was an interesting thing to see come out. And if you certainly scour through the old literature in the early 80s, you'll see stuff about this pop up regularly. So really when the field kind of shifted directions, I would say, was in the late 80s. And the first major study, I think, that really moved the field forward came from Lynn Howlett's lab. And Lynn, who is now in North Carolina, she was a pharmacologist, and basically what they were doing was they were just looking at intracellular signaling responses in cell culture systems of what happened if you bath applied different pharmacological tools. She got into the cannabinoid field, and they started playing with THC and cannabinoid receptor similar structural variants. And basically what they were able to show in the very influential paper that came out in 1988 was the fact that the actions of THC were completely consistent with a receptor-mediated response. And so they could do things like show there was a temperature dependency, which proteins are temperature sensitive, so if you ramp up temperature, proteins destabilize and break down, you lose your effects of the drug. That wouldn't happen if it was a non-specific effect going into membrane fluidity. There was activation of specific signaling transduction cascades that were specific. It wasn't broad, again, suggesting this was a receptor-mediated response. And it was sensitive to various toxins that shut down receptor-mediated signaling. So Alin really generated the most relevant evidence in this sense. And it was saturable. That's another important point, because that doesn't happen unless you're working through a protein interaction. And so Alin kind of created the evidence without identifying a receptor, saying, okay, it's time for us to actually start hunting for this, because there's clearly a specific protein that these molecules are interacting with to exert their effects on cell biology and physiology. So not too long after that, because as technological advancements were happening in the field a lot, and the one thing I will say about the cannabinoid field is it really just followed a lot of the steps of what the opiate field had done. Endogenous opiates had been discovered a few years before, and a lot of the ways that they mapped out the functioning of opiate receptors was through radioligand receptor binding assays and whatnot that were becoming in vogue at the time. And so the cannabinoid field kind of followed this. And this really was Miles Herkenham, who had a lab at NIH in the States. And he had access to, I'm not sure who actually generated the original one, but a CB1 receptor agonist that was radio-labeled. And so what they did is they just did a standard autoradiography approach where they used a radio-labeled molecule that bound to cannabinoid receptors and then looked at its expression across the brain. And basically what they were able to find was the darker the area on that slide is, this is from his original publication, the more cannabinoid receptors there are in that part of the brain. It was very clear that this was a specific response, that you were seeing specific patterns of binding that were region-dependent throughout the brain, and they were reversible using a blocker, like a non-radioactive blocker. So again, this wasn't a non-specific effect. So this was very clear evidence that there was a widely distributed cannabinoid receptor throughout the brain, and that it had specific regional localization. Now the question was, there was no receptor discovered at this time, so looking at Alin's data and Herkenham's data, it was very clear that cannabinoids were binding to a receptor, but nothing had been discovered at the time. So as the rumor goes, Tom Bonner's lab was at NIH down the hall from Herkenham's lab, and they were working on cloning orphan receptors at this time. And at some point, someone walked down the hall with an autoradiogram looking at CB1 receptor binding expression in the brain, at the same time that this lab was looking at an orphan receptor that they hadn't figured out exactly what it was. And basically what they found was when they did their in situ mapping of the gene expression of the cannabinoid receptor throughout the brain, the overlap was quite surprising. And then they went on to, they never formally collaborated these projects, although the two papers did come out relatively close in tandem to each other. But basically what they were able to show together was that this receptor that Herkenham's lab was seeing that THC was binding to throughout the brain was the same receptor that Bonner's lab was actually cloning at the time. And so they named this receptor, once they'd identified it, as being the target of THC, the cannabinoid type 1 receptor. They called it type 1, obviously, because at that point, it was becoming very clear that almost every neurochemical system in the brain signaled through multiple receptors, so there was an expectation they were going to find more in the near future. And so that is just a kind of a crude schematic drawing of what the CB1 receptor looked like once they cloned it. So now this is 1990 we're at now. So, I mean, if you really think of the history of a lot of the neurochemical systems in the brain, like you study things like dopamine, the catecholamines, we're going back to the 40s and 50s. This is pretty new in the field of neuroscience. So because of that, a lot of our understanding of the system is only still evolving now. Okay, so we have our receptor. We figured out that it functions through a receptor. It looks like it's binding to a receptor in the brain, and we've cloned the receptor. So now the question is, like I said in the earlier joke, it's not like we had a receptor in the hopes that humans would find THC. This means there had to be an endogenous ligand that was synthesized in the brain and body that was going to act on this receptor, very similar to how opiate receptors like mu were not built in the body for us to find heroin or morphine, but there was an endogenous ligand like endorphins that actually acted on it. So we go back to Rafi Meshulam's lab now. So he discovered THC in 1964. 1992, Rafi's lab discovers the first endogenous cannabinoid molecule. So two interesting facts about this finding. Number one, the student and trainee in his lab, who was the lead author in this paper, was the same guy who had done his PhD in Alin Howlett's lab. So Bill Devaney was this hugely influential person in the field. He did a PhD with Alin Howlett where he discovers that there's apparently a cannabinoid receptor in the brain, goes and does a postdoc at Meshulam's lab and discovers that anandamide, the first endocannabinoid, actually exists in the brain. And so what was interesting about this is that so they had an idea that this was going to be a lipid, it wasn't going to be a protein, so they went screening for it completely differently. Now, what was funny, and I always try and say this to students, is any significant discovery in science is almost never intended. It's usually an accident. And what we know now is that had this accident that I'm going to say in a second never occurred, we probably would have been a few years behind in discovering the endocannabinoids in the field. And I say this because basically anandamide levels in the brain are generally quite low. Now, what we've learned is that they elevate dramatically in a post-mortal hypoxia situation. So tissue is removed from the body and left out without any oxygen or viability. As those cells start to die, especially in the brain, they start producing mass levels of endocannabinoids. We didn't know that then at the time, nor did they know that the actual native levels of anandamide in the brain were nowhere near detection thresholds for the current level of technology, so they never would have found this. What happened was they were getting brain samples from a butcher, essentially, and they would collect the brains, and in this one experiment, they accidentally left the brain on the bench for the weekend. So it had 2 full days of sitting there in a post-mortal hypoxic state for all these anandamide levels to start accumulating. Come the next week, they took the sample, did the lipid extraction, and that was actually how they ended up finding anandamide. It was completely incidental. Again, one of these things we now know, we never knew then, but it's the serendipity of science I always find fascinating. And so Raffi, who has a very good sense of humor, came up with the idea that, well, you got to come up with a cool name for this molecule. And so he took the Sanskrit word for bliss, being ananda, and then coupled it to, because this was a conjugated version of arachidonic acid and ethanolamine, so it was an amide, to anandamide. And so that's where the name anandamide comes from, for anyone who's interested. It means internal bliss with an amide tail, basically. And so this is what it looks like. And what's interesting is it really doesn't look that much like THC. If you're a structural chemist at all, you look at those molecules and you think that is a strange, you wouldn't really have predicted this would have been the structure. There's enough structural homology for it to bind to the same receptor, although some of the modeling work does suggest that they may bind in slightly different manners. But, fortuitous that we are able to make this discovery as a field and move forward. So again, the 90s were a very exciting time in this field. A lot of massive discoveries were made kind of in rapid fire, back to back. So now we're going into 93. So 1990, the receptor, 92, anandamide. 93 is when we discover CB2 receptors. And this was the Monroe lab that was in the UK. And they were studying the kind of immune system, and they found this peripheral, as they called it, a peripheral cannabinoid receptor, which became termed CB2. And this was found to exist on immune cells. And so they cloned it and published this in a Nature paper in 1993 and showed, okay, now we have a second receptor for cannabinoids to work in. CB2 has been a very tricky receptor for the field to play with and figure out. There's been an unending amount of controversy over whether it truly is a peripheral receptor and is only found on immune cells, or if it is indeed found in brain cells. And I would say that is not resolved yet, so let's keep going. We'll see where the field takes us. But we know it's very highly expressed in immune cells, and it has some very interesting biology to it. So, like I said, the reason why it was referred to as a peripheral receptor is because when they did the in situ screen in the brain, they weren't seeing any receptor levels. Now, I'll talk about this a little bit more later on, but we do know that this is an inducible receptor, at least in a lot of immune cells like microglia in the brain. You don't see a lot of CB2 at rest. What you see is when these immune cells become activated and thrown into a state of inflammation, let's say, the receptor transcript seems to ramp up quite rapidly, and it almost acts as an autoregulatory mechanism on immune cells to gate the amount of pro-inflammatory molecules that are getting released. So it's a tricky guy to chase, the CB2, and I have to say the field's been very interesting, but it's just been fraught with controversy and arguments, which is always fun in an academic sphere. Okay, so then we came across our second endocannabinoid. So this is now 1995. And again, this time it was actually discovered by 2 separate labs in tandem. Raffi Meshulam's back on the scene again. It's clearly understandable why Raffi has established himself as a guru in this field in many ways, because he has driven many seminal findings. But both Raffi's lab and the Sugiura lab in Japan, within a couple of months of each other, I'm not technically sure who had the first published paper, but the 2 papers both came out in 95. Raffi identifying it in the canine gut, and the Sugiura lab discovering it in the brain, but they both found 2-AG. So it's similar to anandamide again, and this is interesting because this is a homology in the system, is these are arachidonic acid conjugated molecules, both anandamide and 2-AG. So their backbone is arachidonic acid, anandamide is conjugated with ethanolamine, 2-AG is conjugated with glycerol. There are some differences, as I alluded to, between anandamide and 2-AG. They do look more similar to each other than either of them do to THC, though. That's obviously the case because of the arachidonic acid backbone. They both, so anandamide, as I'll talk to as I move into the receptor pharmacology a little bit more, anandamide really is preferential at CB1, it seems. It's not a hugely efficacious agonist, but its effects that it exerts on the cannabinoid system do seem to be driven primarily by CB1 receptor activation. 2-AG, on the other hand, does seem to be a full agonist at both CB1 and CB2. So many people, especially Sugiura, have argued that 2-AG is the true endocannabinoid. I don't really see any value in having those debates. They're both signal at the system. They're both relevant for different things. But the fact of the matter is 2-AG is a very efficacious agonist. And we know that if in the brain, for example, you have prolonged exposure to elevated levels of 2-AG, it is far more efficacious at burning out the cannabinoid receptor than, say, anandamide or even THC is. And that's just due to the pharmacological properties of how 2-AG binds and signals through the CB1 receptor. Also, interestingly, its levels in the central nervous system are found at a lot higher levels than anandamide. So 2-AG, in many ways, I can see how the argument has evolved that it maybe is the more primary endocannabinoid, although there's still a lot of relevance for anandamide. But 2-AG is very important in terms of mediating kind of endogenous function of this system. Okay, so we have all our basic players figured out at this point for the most part. We know our receptors, we know our molecules, so we have a feel for how the system is working. Then there was the question of putting in kind of this functional matrix and understanding, well, how is this all integrated and how does it work in the brain? And it was a seminal paper that came out in 1994 from Daniele Piamelli and Vincenzo DiMarzo. DiMarzo, interestingly, is now in Canada, actually. He's at Laval. He's one of these Canada Excellence Research Chairs that's been recruited here. And he's been a big player in the field for some time. But this original paper that they both worked on when DiMarzo was a postdoc with Piamelli was essentially they were able to demonstrate that if you took neurons and cultured them and then stimulated them, neurons had the ability to release endocannabinoids. And so you could detect anandamide if you looked in the, after stimulating these neurons, that anandamide was being synthesized. So what was strange, though, is that the general interpretation of this is that the endocannabinoids were actually getting released from cell bodies because of the way that this prep was set up and just the interpretation at the time. And it was correct, I would say, that it seemed that endocannabinoids were being released. Most neurotransmitters, if you have familiarity with the nervous system and how it works, is they're released from axon terminals. That's kind of the canonical mechanism of neurotransmission in the CNS. This seemed to be a little bit different because it seemed as if the endocannabinoids were being released from cell bodies. So that was kind of, okay, something's a little different here. And as we'll see, as we learn more and more about the field and the system, it really does not play by the general rules that are true of biology of most other neurotransmitter systems. Okay, so now we know that endocannabinoids are released from cell bodies. We know that neurons make them and have the ability to release them. We can detect levels in the brain, and we have receptors that they work on. So the question is, well, what are these receptors doing? And so throughout the 90s, there were a lot of electrophysiology studies that were ramping up because things like patch clamp electrophysiology were becoming more of an in vogue technology at this point. And so you had the ability to isolate individual neurons and look at changes in synaptic currents and inputs. And basically what was established readily from a series of labs, most of this was being done with exogenous cannabinoids, was that if you activate CB1 receptors, the response was a suppression of neurotransmitter release. And this basically meant that activating CB1 resulted in a reduction of the release of neurotransmitter into the synapse. This seemed to be mediated because on axon terminals, where most neurotransmitters are released from, this process, not entirely, but largely involves a calcium-dependent mechanism where you get calcium influx into the axon terminal, and that drives neurotransmitter release. So cannabinoids, one of their mechanism of action, seemed that they had the ability to suppress calcium influx into the axon terminal, and that was at least a relevant mechanism and still has held true in its ability to inhibit transmitter release. Around the same time, antibodies started getting generated to recognize the CB1 receptors so we could start doing more fine-tuned mapping at a microscopic level of where CB1 receptors were sitting in neurons. And interestingly, what was identified through these early studies, it was consistent with the electrophysiology work, was most cannabinoid CB1 receptors seem to not be sitting on the cell body, as most neurotransmitter receptors were, but instead they were sitting on axon terminals. And that was consistent with the ability of cannabinoids to regulate neurotransmitter release. But again, an important finding to help validate that. And that has held true again. The majority of cannabinoid receptors that we know of in the brain sit on axon terminals, and they're very important for gating neurotransmitter release. Okay, so now all the pieces are falling into place, but there was kind of that final thing that was required to tie it all together, which is how does this play out in real time in physiology? So turn of the century, we're into the new millennium now, 2001, and back-to-back, three very relevant and consistent papers came out from three very well-respected labs. Roger Nichols' lab at UCSF technically got the first one. Masanobu Kano in Japan and Wade Reger, who I believe is at Harvard, all kind of had these papers, all looking in different brain regions, where they identified that endocannabinoids actually functioned as retrograde signals. And that made sense based on what we knew at the time. But what I mean by that is that endocannabinoids were being produced postsynaptically in the cell body of a neuron in response to neuronal activation or depolarization, and then they acted as a retrograde signal that instead of going in the canonical axon terminal to cell body, went backwards from the cell body back to the axon terminal. This was really a revolution in neuroscience. And actually, for many people who'd been studying biology for a long time, it wasn't hugely surprising, but it kind of went against dogma. And things that go against dogma are always fun because they open the door to all this other stuff you can chase down afterwards. But at that point, there was a very dogmatic view on neurotransmission that it was anterograde. It came from axon to cell body, and that's how neurons talk to each other. What the discovery of endocannabinoids working as retrograde signals demonstrated was that that conversation goes in both directions. And endocannabinoids were a very important mechanism by which postsynaptic neurons could talk to their afferent input and say, slow down, you're driving me too much, there's too much excitability coming onto the cell, we're gonna burn out, you need to taper down a bit. And in the kind of simplest model, which I'll have a visual for in a second, this is like a circuit breaker. And the idea was, if you looked at excitatory transmission, for example, a lot of excitation would dump onto a cell. That cell would then start releasing massive amounts of endocannabinoids to quench that excitatory drive coming into it, almost as a feedback signal that keeps everything at bay. And that was very important. This has proven to be a very important finding in context of things like epilepsy or other states of hyperexcitability. But this was a hugely relevant finding that tied together all the basic components of what we understood about the field, the molecular architecture of where everything sat in the synapse, and this was basically tied it all together and said, well, this is how it works. And that was very important for the field. So this is a schematic of what endocannabinoids look like and how they function in a synapse. So basically in the top we have our presynaptic axon, and that's coming in, and that's releasing neurotransmitter onto the postsynaptic neuron. This is as good as animation gets for me. And actually I didn't make this, my postdoc did. So this is kind of canonical neurotransmission. That axon's releasing glutamate, it activates postsynaptic neurons. When that activates postsynaptic neurons, we get a calcium influx into that cell. That calcium influx then triggers all the enzymatic machinery that's going to make endocannabinoids. So their synthesis starts occurring de novo. There's not really a storage mechanism that we fully understand. It does appear that endocannabinoids are quote unquote made on demand, where they're synthesized and then used immediately. So as they're made, they then act as these retrograde signals. They travel back across the synapse. They activate CB1 receptors, where that shuts down calcium channels. It also activates outward rectifying potassium channels. Basically what it's doing is quieting down that axon terminal, so that what ends up happening is you get a suppression of neurotransmitter release, and then relatively rapidly endocannabinoids are then degraded and taken offline. 2-AG by this enzyme MAGL and anandamide by this enzyme FAW. So that's kind of a snapshot of how endocannabinoids behave in a synapse and what it is that they do, and what the kind of, let's say, cleanest and easiest to explain model of endocannabinoid function in the synapse goes. Okay, but the discoveries keep going. Well, there's still a lot more for us to learn about the system. Importantly, recently, in the last couple of years, the crystal structure for the cannabinoid receptor was finally resolved and visualized. And this is really important for drug discovery in many ways, because what this shows us, once we have the crystal structures, we start to understand how it is that these molecules dock and bind in the receptor itself. And so this was a really big discovery. It had kind of hampered the field for a while. There's been a couple of follow-ups to this as well to show that the structure, because the structure shifts depending on whether there's an agonist binding at the time or not, and there have been some additions to the original discovery, but it does appear that we're in a good place now with understanding the crystal structure of this receptor. Okay, so it's complicated. This is an understatement, I have to say. I mean, this is almost a very high-level overview, Cole's Notes version of how endocannabinoids function, but it's one of the things I think there are some points that are very important to understand. One of them is neuron type matters. And what I mean by this is the CB1 cannabinoid receptor is really widely distributed throughout the brain. And it is found on a host of different neuron subtypes that exist. So we know, as I mentioned briefly, we have what we call excitatory and inhibitory neurons in the brain. The excitatory neurons will release an excitatory neurotransmitter called glutamate. Inhibitory neurons will release an inhibitory neurotransmitter called GABA. We're discovering some neurons that release both. We're not going to talk about that. But what we do know is that CB1 receptors are found on both of these neuron types, as well as a whole slew of others. So they're also found on dopamine, serotonin, norepinephrine, acetylcholine, neuropeptides. Pretty much every neuron type there is in the brain has cannabinoid receptors. The abundance of them, though, sit on axon terminals of glutamate neurons and GABA neurons. And that's really important because that means, and since we know CB1 receptor activation shuts down neurotransmitter release, this obviously is going to have very different implications depending on which synaptic population of CB1 receptors are being activated by cannabinoids. And so, I can't go back. Maybe I can. Yeah, okay. So think of it this way. If CB1's on an excitatory neuron, if you activate CB1 and turn off that excitatory neuron, you're reducing kind of net neural activity in a circuit because you're reducing afferent excitable drive into neurons. If CB1 is on a GABA neuron and when it gets activated, it shuts down inhibitory neurotransmitter release, it's actually going to excite a circuit by pulling the brakes off and reducing inhibitory tone on that circuit. And so you can understand that the net effect of what a cannabinoid is going to do for a circuit is going to depend very explicitly on which neuronal population of CB1 receptors it's activating. Okay, so here's the circuit breaker model I alluded to before. This is the most, easiest way to explain endocannabinoid function in the brain. It's a circuit breaker, so we get excitatory drive coming from a presynaptic neuron. You get a lot of activation of the postsynaptic neuron. That neuron then makes endocannabinoids, which feed back and act to turn off that incoming neuron. So this is exactly like a thermostat. You set it at 22, temperature rises, heat turns on until it hits 22. Once it hits 22, there's a recognizing mechanism that then turns it back off so that things stay in a very happy place. And in the brain, we want things to stay in a happy place. We don't want them to become too excitable or too quiet because both of those obviously have consequences. Okay, so there's also these heterosynaptic regulators of plasticity. So this means that a neuron can make endocannabinoids and not feed back onto the incoming neuron to turn off excitation, but actually act locally to turn off inhibitory neurons and therefore facilitate activity at a synapse. And so data has been generated to show that they do both. And a lot of this we don't understand why, because we can't visualize endocannabinoids once they get released, really. So we don't know where they're going or what they're doing, but we do know that they seem to have this ability to both feedback on an incoming neuron and shut down excitation in a circuit, or activate local inhibitory or CB1 inhibitory and actually increase the net gain on that circuit. So important to understand the difference there. What's interesting, though, is while it appears that the majority of CB1 is actually on GABA neurons, the functional effects of THC, for example, have almost exclusively been tethered to CB1 and glutamate neurons. And what I mean by that is if you generate mice that lack CB1 receptors on GABA neurons or glutamate neurons, if you delete CB1 and GABA neurons, you lose a huge amount of CB1 in the brain. But if you give an animal an injection of THC, they exhibit all the behavioral indices that they've gotten high. They just kind of lie on the ground. They don't move around a lot. They have a drop in body temperature. All the physiological responses we normally see are intact, which was odd. But you delete CB1 off of glutamate neurons, and pretty much all these effects vanish, which do suggest that CB1 and glutamate neurons is actually very important for regulating the physiological effects of THC, at least the majority of them. Now, one of the things that we've learned about this is that this relates to the ability of CB1 is not a ubiquitous receptor in the sense that it functions differently on different neuron subtypes. And what's basically been discovered is that on glutamate neurons, CB1 is extremely efficient at recruiting intracellular signaling processes, so it's very effective at quenching glutamate release. There's a lot more CB1 on GABA terminals, but it doesn't seem to be as effective at recruiting signaling mechanisms. And there's some unpublished data that should be coming out soon from a colleague that I know in Hungary who does a lot of high-degree resolution microscopy. And basically what he's found is that on the glutamate neurons, CB1 receptors sit right beside where the calcium stores are. And it makes it very accessible to rapidly modulate neurotransmission, whereas on the GABA neurons, they're very kind of stochastically spread all over the axon and the axon stalk, not even in immediate vicinity of the calcium stores. So you need a lot more gross activation of CB1 on those GABA neurons to really modulate the GABA signal. So this would explain why low doses of THC and the prototypical effects of intoxication, like the changes in body temperature, pain sensitivity, increased feeding, reduced anxiety, all of these effects are all mediated by CB1 and glutamate neurons. because those neurons are going to be more exquisitely sensitive to low doses of THC. The thought is, as you amp up THC concentrations or higher dosing, the reason why there's a greater preponderance of adverse effects where people get panicky or paranoid or have differential outcomes is probably due to this kind of pleiotropic effect where you start activating CB1 broadly on a whole host of neuron subtypes. And that's going to vary from individual to individual because there's a huge amount of individual variance in terms of CB1 receptor expression in the brain. There's also a developmental component. I'm not going to talk too much about this, but basically we do know that the system evolves in babies and in utero and in young children. This system is rapidly evolving. It has a lot to do with neuronal migration and circuit formation in the brain. And somewhere as you go into juvenile development, the system flips and becomes much more important for regulating neurotransmission. But what's also interesting is that its ability to regulate neurotransmission changes with age. So around the adolescent window, it's far more effective at regulating glutamate. And as an organism ages beyond puberty and into adulthood, a lot of CB1 starts dropping off those glutamate neurons and having a higher expression on GABA neurons proportionately. And it's interesting because if you look at the patterns of cannabis use in the general population, 18 to 24-year-olds almost always represent the largest catch of group of people who are using cannabis. And a lot of people who smoked cannabis as a teenager or as a young adult, as they get older, this wanes. And surveys that have asked them why, what was your motivation for the change in pattern? Certainly some of them, you know, we want to be adults and grow up and stop doing things we did as teenagers. But a lot of people actually report that they said, the thing I've seen repeatedly is this quote, cannabis turned on me. I used to enjoy it when I was younger, and as I got older, I found it became more adverse. And I find that really interesting because of the way the system evolves across age span. And so it's not going to be true for everyone, obviously, but for a large proportion of people, it does appear that things that used to be pleasurable relating to cannabis have become adverse with age. Sorry, I gotta just run through this. Okay, so we're also outside of the brain. Cannabinoid receptors are basically in every biological system in the body. We have them in the liver, the adipose tissue, the GI tract, the muscle, the pancreas. And there is both, this is, again, beneficial and potentially not beneficial depending on the outcomes, but you can see there's a lot of different layers through which the system can modulate physiological function in the body, and through its processes and all these different tissue depots, there is both opportunity for therapeutic intervention for various outcomes and disease states, but there's also the possibility of having interactions with other systems. Okay, so cannabis in the endocannabinoid system. So very quickly, because I don't want to run too over time and I'm getting close. Like I said, one of the things that I appreciate being a basic scientist who works in the system is it kind of takes a lot of the creativity out of it, so it's kind of fun. You can just say, well, what's an effect of cannabis consumption, and then guess if that has a role in a physiological process. And that was the way a lot of the field behaved in the early days. We have to be more creative now, but when it was first discovered, it wasn't too hard to start guessing what processes were going to be related by endocannabinoids. You know, the most prototypical effect people report of smoking cannabis or consuming cannabis is the munchies. Not surprisingly, endocannabinoids are an important regulator of feeding behavior. They drive feeding behavior, especially after periods of fasting. Endocannabinoids, similar to the way endorphins are, we know that their release is provoked by noxious stimuli, like painful stimuli, and they act as a feedback signal to help dampen pain sensitivity, so they have analgesic component. Things like stress-induced analgesia, for example, we know, seem to heavily involve an endocannabinoid component. And again, not surprising, because for centuries, people had been using cannabis as an analgesic themselves. And for the purposes of what my research is, what I found interesting was the fact that a lot of people, as I'd alluded to, the majority of people who smoke cannabis on a regular basis do so through self-report because they say that it helps reduce anxiety and stress, it helps them sleep at the end of the day. I kind of look at it as the equivalent of someone having a glass of wine or a nightcap. That seems to be a common reason. It's to take the edge off at the end of the day. And it's interesting because a lot of the work that we've done now, not just our lab, but lots of labs, has clearly demonstrated endocannabinoids do have an important role in stress recovery, termination of the stress response, and adaptation to stressors. So this is interesting because, again, what we know about the effects of cannabis and the motivations for use of people have directly mapped onto what we understand about the physiological processes of endocannabinoids. This is an image, actually, that the CCIC has made, so it's easy to use in a venue like this, although there's copyright credit given at the bottom. But this is just kind of a human brain image of that initial autoradiogram from the Herkenham lab that I was showing, in the sense that The darker the green, the more CB1 receptors there are in those areas. And as you can see, it's really widely distributed throughout the brain. Areas like the hippocampus, which are really important for memory consolidation, among other things. There's a huge density of CB1 receptors there. It's found in feeding circuits. It's distributed throughout the cerebral cortex, the cerebellum. So again, motor effects, a lot of these other effects are not surprising based on where we see CB1 receptors. And this, I believe, Mark, is this on the website still? Yeah, so this image is on the website if you're curious. This kind of is a nice snapshot about what we understand about the neuroanatomy of cannabinoid receptors and what they regulate. Okay, so let's not forget about CB2. I always get asked about CB2. It is a very interesting receptor, as I alluded to. As I said before, the majority of data still confirms that CB2 is primarily an immune-based receptor. And what I mean is it's primarily found in immune cells. This is like macrophages, B cells, T cells in your periphery, but also microglia in the brain. Activation of CB2 functions primarily to suppress the release of pro-inflammatory cytokines. That also modulates for peripheral immune cells adhesion molecules and can prevent cell tissue entry. And so a lot of immune cells will have this rolling process where they roll along the inside of the vasculature through adhesion molecules, and they can infiltrate different tissue depots in the body. This process, though, of those adhesion molecules, that can be blocked by CB2 receptor activation, as can the release of pro-inflammatory molecules. And again, as we understand more and more about the biology of the cannabinoid system, it's not surprising when you look at a lot of the medical issues that people use cannabis for. Inflammatory diseases across the board are one of the most common classes of disease that people use cannabis for in a medical domain. Things like arthritis, MS, IBS. There's a whole host. Obviously, you're relatively familiar with that in the audience here. But it is very interesting about how cannabinoids have this very specific effect through CB2 on immune cells. What's interesting, and this is what I alluded to, and this was actually some original work I believe was done by, the initial discovery of the fact of this was found by Donald Abrams, I believe, in San Francisco, because they were looking at using cannabis as a means to treat wasting syndrome and cachexia associated with AIDS. This was in the 80s during the AIDS epidemic. And so they thought by using this they could help restore feeding and get people to eat so they wouldn't waste away and you'd have a greater survival rate. But we knew that there had been some documented effects of cannabinoids on immune function, so there was a concern that in an immunocompromised state that administering cannabinoids would have a further damaging effect, since they seem to be kind of anti-inflammatory and suppress a lot of immune processes. What was really interesting is they didn't find any evidence of that in the original populations. And as we've understood more and more about the system, this makes sense, because immune cells at rest don't really seem to express a lot of CB2 receptors. What you actually seem to see is that it's an inducible receptor. So this is especially true on microglia in the brain. There's almost no detectable CB2 at rest. An inflammatory event happens, like an infection or something that causes inflammation in the brain. Those microglia kind of wake up. They go from a surveilling state to an active state where they're trying to clear pathogens from the brain. And in the process of doing this, they also start secreting a lot of inflammatory molecules. And at the same time, they ramp up CB2 receptor expression. So these microglia now have a receptor that is responsive to cannabinoids that helps calm down the release of these inflammatory molecules. Now, this is really important when we start to think about mechanisms of inflammatory diseases, especially neuroinflammatory diseases like MS, and how it is that cannabinoids could play in here. So that's an important thing to know there. Okay, so the general gestalt of what endocannabinoids do in the brain, I would say, I'm not saying this is true for cannabis, but I'm saying this is kind of where the field has figured out one of the main roles of endocannabinoids. I look at them as kind of a vanguard of homeostasis. So we want everything to exist in this very happy range, and when something happens and it disturbs our basic physiology and we move out of that range, one of the jobs of endocannabinoids is to pull us back. So they act to keep things at rest and keep them living in a happy range. If we get stressed out, we make endocannabinoids, they calm us down, they bring us back down. We're starving, we want to eat, endocannabinoids go up, they make us eat, we eat, they go back down to normal. So they're very responsive to external circuits. And as I said, when it came to things like excitability in the brain, we know endocannabinoids are there. A lot of their main job is to protect the brain. The reason that the initial discovery of anandamide happened, because they left the brain tissue on the counter for 2 days, was because as neurons start to die, one of the processes that they do is there's this spreading damage that happens, because as a neuron dies, it releases a lot of calcium and it triggers kind of an excitotoxic spreading neuronal death to other areas. This is one of the reasons where something like a stroke or a brain trauma happens. You have an infarct area or an area, and then you have this kind of spreading damage that occurs after the fact. And that spreading damage is due to this excitotoxic spread. And one of the main roles of endocannabinoids is to quench that and prevent that spread as much as possible. So they're acting to kind of protect the brain and reduce that amount of excitotoxicity. CB2 is acting to suppress inflammatory processes. So they go up, they have a benefit. Inflammation in an acute phase response is important for us. We want it there, but we don't want it to stay on for a long period of time. Endocannabinoids are pulling us back. And this seems to be similar with respect to aging when inflammatory processes kind of progressively elevate and a lot of other things happen. Endocannabinoids are trying to keep things at bay. So there has been a few papers in the last couple of years that have suggested that low dosing of THC or boosting of endocannabinoids may have some protective benefit in the aging brain. Well, I think we'll have to do a lot more research in that to be confident in that area. But okay, so I'm just going to end. I think I have 2 slides left. Hopefully I'll run through these. Where are we? Okay. So targets of phytocannabinoids. So the 2 main cannabinoids that we study in cannabis, as I've alluded to, we have THC, which looks like that, which if you remember what anandamide looks like is not similar at all. And then we have cannabidiol. So these are the 2 main cannabinoids that have received the most research-intensive focus over the last few years. So what are they doing? Well, THC we know primarily exerts its effects through CB1 receptors. And when we talk about the prototypical effects of intoxication, at least using animal models, those seem to be primarily mediated by CB1 on glutamate neurons explicitly. CBD is very unclear. It does not seem to bind to CB1 or CB2 in the standard manner of being a true kind of agonist or antagonist. There has been some recent work in the last couple of years that suggests that might be an allosteric modulator of the cannabinoid receptors. That's only going to mean something to anyone who's in pharmacology. But basically it means it doesn't bind to the normal site, it binds to another site on the receptor, and it can modulate the activity of the receptor. So there is possibility that CBD could influence cannabinoid receptor activity, but it's not a standard agonist. We know it does do a lot of other things. It inhibits adenosine uptake, so it promotes adenosine signaling. I refer to this as the anti-caffeine, because caffeine does the exact opposite. And so this is consistent with some of the sedative properties that have been established about CBD, because it's enhancing adenosine, which is also an inhibitory transmitter. It's an antagonist at GPR55. This seems to be relevant for the anti-epileptic effects of CBD. Some recent work from Bill Catterall and Effie Stella have a nice paper suggesting that the ability of CBD to block GPR55 is why it is effective in Dravet syndrome. We know it activates serotonin 1A receptors. I know there's a couple of people from Linda Parker's lab here today. Linda Parker at Guelph has done some work with this, especially looking at how CBD acts on 1A receptors to promote actions of serotonin in some regards. That may relate to some of the anti-anxiety, antidepressive quote-unquote effects that have been seen with CBD. Also, it inhibits FAW, which is the enzyme that metabolizes anandamide. So there may be this indirect ability to augment endocannabinoid signaling by boosting anandamide. And last slide is a nightmare to look at, but this is just to point out to you, all I've talked about really is cannabinoids, CB1, CB2, and mostly THC, a little bit of CBD. This is everything else. This is only touching the surface now of some of the other molecules that are found in cannabis, but you can start to see the diversity in the mechanisms through which these molecules can interact with a whole host of biological systems to modulate a huge amount of physiological processes. And so this is why I'm just going to end on the one note I think is very hopeful. Moving forward now through a legalization framework in Canada, we can really be a leader in the field, I think, to start understanding biological variants in cannabinoids coming from cannabis, therapeutic potential, understanding how these molecules work, and moving forward in the field. So thank you for your attention. Everyone seemed to stay awake, so that's a plus. Thanks a lot.